Hello everyone, I am Vijay Kevlani, Assistant Professor, LG Institute of Pharmacy. Welcome to Pharma Ignite. So you already know that we have started online lecture series to utilize this time of lockdown. I wish you will also utilize this time to learn these new things. We are giving our full efforts to make this kind of videos for you. So please utilize this time and learn the things you are going to have in your syllabus in this semester. Right? So without further ado, let's move on to our today's session, that is pathophysiological role of histamine. So let me remind you what we have studied so far. Very first is we have started with introduction of autocoils. Then we started histamine. In that we studied turnover of histamine, receptors of histamine. Lastly, in last series or last lecture, we have studied pharmacological actions of histamine, and in today's session we are going to do this pathophysiological role of histamine. Now let me tell you what is pathophysiological role. So pathophysiology, we already know that physiology means normal function, while pathophysiology means altered function, right? Altered function of any tissue, altered function of any organ system. That is pathophysiology. So what histamine is playing role in alteration of this function of specific organ system, that we are going to study in today's session. So pathophysiological role of histamine. Very first is gastric secretion. So histamine has dominant physiological role in mediating secretion of HCl in stomach. So as you can see that histamine is present in our stomach, right? It is non-mast cell histamine. Okay. Now here in structure of parietal cell, you can see that there are histaminocytes present nearby. This histaminocytes will secrete this histamine, right? And this histamine is going to bind on H2 receptor present on parietal cells. Now what will happen? So ultimately this H2 receptor will stimulate proton pump and it will stimulate HCl secretion. So that is basic mechanism of HCl secretion. Now what happens whenever there is excess HCl secretion? It will cause peptic ulcer. So what actually happens here, that histamine is released from histaminocytes under certain stimuli, like whenever there is consumption of food or whenever there is cigarette smoking, alcohol. This kind of stimuli will cause release of acetylcholine from vagus nerve, right? Ultimately vagus nerve is stimulated. This will release acetylcholine, and under influence of acetylcholine this histaminocytes are stimulated. Now this histaminocytes will release histamine, and this histamine is going to bind on H2 receptor present on parietal cells, right? Now by that ultimately parietal cells are stimulated, and specifically proton pump present on parietal cell is stimulated, and it will secrete H plus, and thus it increase HCl secretion. Once again, very first is any stimulus that release acetylcholine. This acetylcholine will stimulate histaminocytes. Histaminocytes will release histamine, which is going to bind on H2 receptor, and then ultimately there is stimulation of parietal cells, which will cause HCl secretion. Allergic phenomena. So mediation of hypersensitivity reaction was the first role ascribed to histamine. It is an important but only one of the mediator of such phenomena. Released from mast cell following antigen antibody reaction on the surface in immediate type of hypersensitivity reactions, histamine is causative in urticaria, angioedema, bronchoconstriction and anaphylactic shock. Now what does it mean? So what happens, as we already know that Dale studied this histamine in 20th century, and he found that there is a close relation between this antigen antibody response as well as injection of histamine, right? So whenever histamine is injected, the responses are produced. These responses are same which are produced under antigen antibody reaction, or you call it allergic reaction. So that proved that histamine is having certain role in allergic reactions. But yes, please remember that it is not only one mediator. So moving on, the H1 antagonists are effective in controlling these manifestations to a considerable extent, except asthma and to a lesser extent anaphylactic fall in blood pressure, in which leukotrienes appear to be more important. Now, what does it mean? So, H1 antagonists are useful whenever there is histamine release. In allergic condition, there is a release of histamine. That's why we can use this H1 antagonist. But in asthma, more than one mediators are playing role. So in asthmatic condition, histamine antagonists are not sufficient, right? Secondly, in several anaphylactic attack, right? In that condition also, leukotrienes are playing important role. That's why histamine antagonist will not work. Moving on, third one, as a transmitter. So histamine is believed to be the afferent transmitter which initiates the sensation of itching and pain at the sensory nerve endings. Non-mast cell histamine occurs in brain, especially hypothalamus and midbrain. In the brain, H1 agonism suppresses appetite. This may explain the appetite promoting action of certain H1 antagonists. Histamine also appears to participate as a transmitter, regulating body temperature, cardiovascular function, thrust, and possibly other functions, mainly through H2 and H3 receptor. So, as we know that histamine is present in our brain, mainly it is present in hypothalamus, and apart from that in reticular activating system, which maintains our wakefulness, right? So, whenever we are awake, the neurons of this reticular activating system are releasing histamine and generating action potential, right? They are stimulating each other, and that is why we feel awake. Whenever these neurons are blocked, whenever the action potential is not generated properly or is generated less, right, than normal, at that time we feel sleepy, right? And that is what these drugs of flu are doing, right? Whenever we take this anti-allergic drugs, ultimately what they will do, they will cross blood-brain barrier, they will go to our this reticular activating system, they will block H1 receptor there, and by that it may cause sleepiness, right? So that is basic mechanism behind it. Apart from that, histamine is also controlling our body temperature, thrust, and many other functions, right? Moving on, inflammation. So histamine is a mediator of vasodilation and other changes that occur during inflammation. It promotes adhesion of leukocytes to vascular endothelium by expressing adhesion molecule P-selectin on endothelial cell surface, accumulating leukocytes at the inflammatory site, right? So what happens at the inflammation site ultimately? Initially, there is a release of histamine. This release of histamine will stimulate endothelial cell to express P-selectin. Apart from that, histamine will act as a chemotactic agent, and it will accumulate or it will attract this leukocytes, and it will cause addition of leukocytes to this P-selectin molecule. So in that way, it is playing role in inflammation, right? There are certain other mediators also which are playing important role in inflammation, like cytokines, right? Fifth number, tissue growth and repair. Because growing and regenerating tissue contains high concentration of histamine, it has been suggested to play an essential role in the process of growth and repair. So whenever after trauma there is growth, histamine is present. It is found that histamine is present after trauma at the site of injury. So it may promote growth. It is not fully accepted, but still it is one of the point that it may promote growth of that tissue. Moving on, headache. So histamine has been implicated in certain vascular headaches, but there is no conclusive evidence, right? So as we know that histamine is vasodilator. So whenever there is release of histamine in our brain, It may cause vasodilation of cerebral blood vessel and that may cause headache, but there are no certain evidence present, right? Next is uses of histamine. So histamine has no therapeutic use. So far it is not used for any therapeutic process due to its effect ultimately. In the past it has been used to test acid secreting capacity of stomach, bronchial hyperactivity in asthmatics, and for diagnosis of pheochromocytoma. Pheochromocytoma is tumor of adrenal gland, right? But this pharmacological tests are very risky and not used nowadays, right? Beta histine. So beta histine is an analog of histamine. It is an orally active, somewhat H1 selective histamine analog, which is used to control vertigo in patients of Meniere's disease. Now, Meniere's disease is a disease of inner ear. It is related to vestibule, right? So, whenever there is damage to vestibule, it will cause vertigo and spinning, right? So, to treat that, analog of histamine, that is beta histine, is used. It is possibly act by causing vasodilation in the internal ear. It is contraindicated in asthmatics and ulcer patients. So as we know that histamine is ultimately producing this vasocontraction as well as smooth muscle contraction. So whenever if asthmatic patient will take this kind of drugs, there would be a contraction of bronchi and it may aggravate the condition of asthma. Secondly, histamine also induces acid secretion. So whenever someone is suffering from peptic ulcer, what happens ultimately? Histamine will cause increase in acid secretion. So that person should not take this drug, right? Histamine release. So we have already seen that how histamine is released after antigen antibody reaction. So in this part we will see how this histamine is released due to other factors, right? A variety of mechanical, chemical and immunological stimuli are capable of releasing histamine from mast cells. Very first is tissue damage. So due to trauma, stings and venoms. Apart from that, release of proteolytic enzymes and phospholipase A. This may cause tissue damage and ultimately cause histamine release. Antigen antibody reaction involving immunoglobulin E antibodies. Third is polymers like dextran, polyvinyl, pyrrolidone. Fourth is some basic drugs like tubercularine, morphine, atropine, pentamidine, polymyxin B, vancomycin and even some antihistaminics release this histamine by displacing it from the binding site and not by immunological reaction. So now what does it mean? So ultimately, if you have studied general pharmacology, there is displacement topic under distribution of drug, right? So what happens ultimately? Every drug or histamine is bound to specific site of tissue, right? And this drugs may displace this bound histamine and ultimately by that it may increase concentration of histamine in plasma, right? It is not immunological reaction, it is drug-drug interaction, or you call it pharmacokinetic drug-drug interaction. Moving on, surface acting agents like twin 80 and compound 48 by 80. The primary action of this substance is release of histamine from mast cell. Therefore, they are called histamine liberators. Now, this definition or this word is too much important. Histamine liberators. So, what happens actually? So, as we know that so far we have studied there is a release of histamine, but that histamine is non-mast cell histamine. This substance are specifically releasing mast cell histamine and that is why a different term is coined for this substances, that is histamine liberators, right? It may be asked in 1 marks. They produce an anaphylactoid reaction like itching, burning sensation, flushing, urticaria, fall in blood pressure, tachycardia, headache, colic and asthma, right? Thank you everyone. Hope you like this session. Please like, share and subscribe our channel Pharma Ignite. Thank you.